Okay, so this is the start of B2 past papers I'm going through. Obviously, I'll give you extra information where it's needed. So this will be a revision guide and answers and how to answer the questions from the exam paper. So first, we're going to be doing the 5th of November 2012. I hope you are all okay. And we'll just so turn to the first page and have a go at the questions, and then I'll go through the answers with you. So this is your chance to pause the paper or the video and have a go. So pause right now. Okay, so first I'm going to go through the answers. Okay, so it says when a human body cell divides by mitosis, two diploid body cells are produced. Each of the cell contains identical sets of chromosomes, and which are found in the nucleus. Right, mitosis occurs during growth and repair, and also asexual reproduction in bacteria. Right, the final answer rather, is why are embryonic stem cells important in research? Because they are undifferentiated cells. This means that they can become any type of cells, so it's particularly important with people who need replacement organs or who need specialized cells. So you can take the embryonic stem cells and make it into any type of cell, and so that's quite useful for people with particular diseases. If they need a new liver, then they can just use the embryonic stem cells to make a liver, and then that person will be happy. So I'm going to look at these keywords and see what some of these keywords mean. Okay, so cytoplasm is where the chemical reactions happen in a cell. A diploid is when you're talking about the full number of chromosomes in a nucleus. So the diploid number for human is 46 chromosomes, because you get 23 from mom, 23 from dad. That, if you add them together, that's 46. That's your full set of chromosomes found in every single cell of your body except your sperm cell and your egg cells. Okay, we'll talk about that in a bit though. But every other cell in your body has 46 chromosomes, and of course the red blood cells has 0 chromosome because it does not have a nucleus. Okay, so that's diploid. Diploid is the full number of chromosomes. Next we've got haploid. So haploid is half the total number of chromosomes in your nucleus. The only cells that have haploid number of chromosome are sperm cells and egg cells. Okay. The nucleus is the control. It controls the activity of the cells. It contains DNA, and DNA codes for proteins. Okay. So that's that. And like I said here, growth. So mitosis occurs during growth and repair of cells, and also asexual reproduction in bacteria. So turn over to the next page, have a go, and then I'll go through the answers. So the answer to the top question is 120 minutes. This is how to work it out. So at time 0, you've got one cell. Now you got to remember is mitosis is you're making 2 cells each time, so you're doubling the number of cells that you have. So at time 0, you've got one cell. 30 minutes later, you've got 2 cells. Another 30 minutes later, you're going to double these, because remember it's dividing each cell by 2. Right, so another 30 minutes you're going to have 4 cells. Another 30 minutes later you're going to double the amount of cells, which is 8 cells, and finally you're going to double those 8 cells again, and you'll have 16 cells. So if you add all the time, for each time it doubles to get to 16, you'll have 3, 6, 9, 120 minutes. So 120 minutes is the correct answer. Next, we're going to have a look at this. So it says organisms that are genetically identical are called clones. Okay, so they have no genetic variation, they are identical, they're called clones. Gametes are sex cells. Sex cells include sperm and egg cells. Okay, and in plants they are pollen and egg cells. Okay, so pollen is the male bit and egg cell is the female bit. And once you have these sex cells fertilized, it produces zygote. Okay, so that's what a zygote is. It's a fertilized sperm and egg cell. Okay, so now what I'm going to do is I'm going to explain mitosis and meiosis, okay, because it's one of the more difficult topics in biology. Now, mitosis, as we've already spoken about, it includes growth and repair. So when you're repairing your own body cells, that's what happens, and when you're growing, you have mitosis, right? And also, it's involved, as we said, in asexual reproduction in bacteria. Now, another word for bacteria is prokaryote. You should know that from B1. So what we can do is we can take how mitosis works. Is, let's say we've got a diploid, a chromosome number of 14. So this cell has 14 chromosomes in it. Now what happens is, number one, we have replication. Okay, so the number of chromosomes that you have doubles. So that's 28 you've got now. So that's the first phase of mitosis. You've got replication. Then the next phase of mitosis is cell division. And what you'll end up having is you'll have 14 chromosomes in one and 14 chromosomes in the other, which is exactly the same as what you had in the parent cell. So in other words, what you have is you have one diploid cell going to produce two genetically identical diploid cells, and these two that are made at the end are called daughter cells. Okay, so you got two identical daughter cells. They are identical to each other and they are identical to the parent cell. And so what we can say is that in mitosis you have no genetic variation, so you've got clones being made. And a problem with this is that you might inherit weaknesses. So if this one had a weakness that it wasn't resistant to a particular drug or something, these are also going to have that weakness because there's no variation at all. So just to recap mitosis, you've got your parent cell that has a diploid number of cells, so it's got the full number of cells. It replicates, so it doubles its DNA content, and then it divides it into two cells that are exactly the same as the parent cell. Now here is meiosis. Now, to remember meiosis, the E in meiosis, you can just write sex there, and meiosis is about sex cells. So this is the production of sperm and egg cells. So what you have is within humans, we have 46 chromosomes total. So that's our diploid number of cells, because that's the total number of chromosomes that we can have. So we have the same thing happening again. We have replication. So it replicates, it doubles the original amount. So we'll have 92 chromosomes in total. But here where it gets different. Okay, within meiosis we have crossover of genes. So the genes swap places. And this is why we have variation. It's the reason why every single one of us looks different. It's because our genes have been crossed over in different combinations. Then what happens is you have cell division, and you'll have 4 haploid cells. So if you look here, we have a diploid cell, which is the full number, 46, and by the end of meiosis we'll have 4 haploid cell, which is half the number of chromosomes in each cell. So we started off with 46, half of 46 is 23, right? 23, 23, 23. But the question is, where did the extra, so this is 46, the question is, where did the other 2 come from? And the answer to that is from the doubling. Once we doubled it, you half it again, and so we're halving 92. So 23 add 23 makes 46, 23 add 23 makes 46. You add 46 and 46 together, you'll end up with 92. Where did that 92 come from? It came from the diploid, which ended up replicating, which ended up forming haploid cells. So these could be sperm cells or egg cells, because it's to do with gametes, which are sex cells. So you form you start off with one diploid cell, and it turns into 4 haploid sex cells, which are aka gametes. So pause the video and have a go at the next question. Okay, so I'm going to go through this question and the next question on the same video. So if you haven't done that, you can pause it and have a go, or you can just carry on with me. So this is a microscope, okay, and I want to go through each bit and what it does. So the eyepiece aids in magnification, okay, but the real magnification comes from the objective lens. the objective lenses. Okay, so you can increase and decrease the magnification depending on which objective lens you're using. So if you're using a objective lens with great magnification, you're going to have greater magnification, obviously. If you're using an objective lens with less magnification, you're going to have less magnification. The stage is where you put your slide. The mirror, this mirror here, reflects light into the microscope, and because this is using light, we call this a light microscope. And the focusing wheel, what this does is it produces a clearer and detailed image. So you can kind of adjust this to make the image more clearer, more focused, and so you can see more detail depending on how clear you can get it. Therefore, the answer is D. Okay, so turning over, we're going to have a look at this. I'm going to do this question first. So it tells you that the length of this cell before it's magnified is 0.05, and it's used a magnification of 400 times. So if you think about this formula, right, it makes sense if you think about it. To get the magnified length, right, so how much it's been magnified by, you take the length that it is at the beginning and you multiply it by the magnification. Just think about that for a second. To get the magnified length of anything in a microscope, you take the length that it was at the beginning and how much you magnified it by, and you multiply them together. So that's what I did. You took the length at the beginning, multiply it by its magnification. It's 0.05 times 400, which gives you 2 millimeters. So here it says, state the function of a mitochondria. Now the mitochondria is up here. I've written it over here. It's involved in respiration, and its job is to release energy. Now, because we've said the word respiration, we need to talk about that equation because it's very, very important. Respiration is glucose plus oxygen, which makes carbon dioxide plus water, and in brackets I've written it releases energy. Okay, brilliant. So let's go through each one of these parts of an animal cell. Now, number one, I know it's an animal cell because it's got no cell wall. I know it's not a bacteria cell because it has a nucleus. So you guys still have to be thinking about the stuff you learnt in B1. So anyway, a nucleus, it controls the activity of the cell. It contains DNA, and DNA codes for proteins. Okay, so that's kind of a bit of extra information there. But this is what you need here. But you need this because they always ask that. Next, cell membrane is it controls what goes in and out of a cell. So this is like the door. So it controls what goes in and out of the cell. That's the job of the cell membrane. And we've already mentioned the mitochondria. I just drew this in because it's important. The mitochondria is where respiration occurs. The equation for respiration is glucose plus oxygen makes carbon dioxide plus water, and it releases some energy. Yeah, brilliant. Okay, so I'm going to upload this video and then I'm going to do the next in the next video.